and welcome to this video by Get Ahead Institute. And in this video, we're going to cover gametogenesis, the first part being spermatogenesis. So if you have not subscribed to this YouTube channel, kindly do so and leave a like as well to help with the YouTube algorithm. So let's get right into it. So, uh, gametogenesis basically uh, is formation of the gametes. Okay, from the word gametogenesis, gameto, formation of gametes, and then genesis, that's the beginning, origin of something. So we're talking about the origin of gametes, basically. So, uh, gametogenesis, by definition, textbook definition, is the process by which sex cells, because those are what gametes are, sex cells divide and differentiate. Okay, these processes prepare sex cells for differentiation, and we have two types of gametogenesis, which is spermatogenesis that occurs in males and oogenesis that occurs in females. So the gametogenesis that occurs in males is called uh, spermatogenesis because it's formation of sperms, while gametogenesis that occurs in females is called oogenesis because that's where oocytes are formed. So in this video, we are particularly dealing with the first part, or the part that happens in males, which is uh, spermatogenesis, and it's going to be a very, very short tutorial. Okay, so before anything else starts, you have what are known as primordial germ cells. These are the cells that remain in the gonads. Okay, let's call them gonads for now. Uh, which are the origin of the gametes or the sex cells themselves. So these divide by mitosis to increase in number, and then they divide by meiosis to produce the gametocytes. So as they are differentiating, there is what is known as cytodifferentiation. Okay, so what we're basically saying here is that primordial cells, primordial germ cells or PGCs, are cells that are contained in the uh, in the sex organs of individuals, males or females, and these are going to divide two ways. One by mitosis to increase their number, and then two by meiosis to produce gametocytes. So these ones are not the actual the actual sex cells, but they are the origin cells through which you get the gametes. So they are going to occur by mitosis, for example. So that is since this is not reductive division, they are going to divide normally just so that they increase in number. And after increasing in number, they are going to divide by meiosis, which is a reductive division from the previous video we mentioned. And they produce the gametocytes, which are the sex cells themselves. So the kind of division that they undergo to make sure that there is a constant production of sex cells in males, particularly. Okay, because in females, it's quite different as we will explore in oogenesis. But in males, there is what is known as asymmetric cell division. Asymmetric cell division. This is where one daughter cell remains as a primordial stem cell, while the other proceeds into the next cell type or cell line. And this was explored in the previous video. So what that means is that at the end of the day, you're going to be having a constant supply of new cells while the primordial cell population still remains. Okay, that's what is happening there. So spermatogenesis, to now dial it in, is basically the distinct sequence of events by which spermatogonia are transformed into mature spermatozoa. So from the progenitor germ cells, which are the PGCs, once one of those cells, so we have a PGC, basically a PGC here would differentiate into two daughter cells. A PGC would differentiate into two, okay, I don't know why I'm not able to write, but would differentiate into two daughter cells. One of them is going to remain as a PGC, and then one of them is going to commit to become a sex cell. So once this one commits, it to now be called a spermatogonia, spermatogonial stem cell. So you can call them spermatogonial stem cells or spermatogonia themselves. So once the spermatogonia uh, are produced, those processes starting from the spermatogonia to the production of the full mature spermatozoa, you are calling that spermatogenesis. Okay, so this begins at puberty from age 13 to 16 and it continues until old age in a man. Okay, primordial germ cells give rise to spermatogonial stem cells, like I said, and spermatogonial stem cells are going to give rise to spermatogonia that are now going to undergo spermatogenesis. Okay, so this is the arrangement of these cells in pictorial form. If you look at the testis of a man, you're going to find this arrangement of cells that aids in the production of the germ cells, the spermatozoa themselves. So you have different types of cell lines that will only make sense after we have covered the length of this video or the length of this topic. Okay, so spermatogenesis, after the spermatogonia have been produced, we're going to have type A, okay, type A spermatogonia, which are going to divide. Okay, so we're at the top here. We have type A spermatogonia that are going to divide into type A pale spermatogonia. So the first type is dark, and the second type is pale. And then the pale spermatogonia are going to divide again into another set of pale spermatogonia, which are going to divide into type B spermatogonia. So type A, dark, pale, and everything, all of those are just ways of increasing in number, like the spermatogonia themselves are trying to increase in number. Where does reductive division actually occur? It starts at the primary spermatocytes. Okay, so spermatogonia all the way up to here, these cells are just increasing in number. But then the primary spermatocyte starts meiotic division. Okay, so type E spermatogonia divide into primary spermatocytes, and then the primary spermatocytes are the first cells that undergo meiotic division, that is meiosis 1. So meiosis 1 happens in the primary spermatocytes, and then the meiosis that we talked about in the previous video about cell division, once that occurs, it's going to give rise to secondary spermatocytes. Secondary spermatocytes are going to undergo meiosis 2, that second type of meiosis. Then the number of the DNA that you're going to have for secondary spermatocytes is now going to be reduced. Okay, the DNA number is going to be reduced. You're going to move from haploid, or rather from diploid to haploid. Okay, so primary spermatocytes, those are going to be diploid, while secondary spermatocytes are going to be haploid. So they can ask you which cells first undergo meiotic division, or the first meiosis, that's primary spermatocytes. The cells that are first to be haploid in the cell line for spermatogenesis, you are going to have the secondary spermatocytes. So these secondary spermatocytes are further going to divide. Okay, they are further going to divide, and or rather, this is no longer called division at this point. It's rather specialization. It's rather differentiation. Okay, so there is what is known as incomplete cytokinesis from the point of a dark spermatogonial cell dividing all the way up to a late spermatid. Okay, all the way up to a late spermatid. What we mean by incomplete cytokinesis is that although these cells are dividing like this, what you can see is that they're still attached by a single point there. Okay, that is incomplete cytokinesis. What does this do? Some research material will tell you that it's responsible for helping the cell exchange nutritional contents, support, and everything, but that has not really been conclusive. But that's basically what is happening there. So from there, like we say, we have secondary spermatocytes that now undergo meiosis 2 to become early spermatids that are haploid, and The early spermatids are the ones that are going to be uh, uh, to have the DNA amount of N. So I did explain this in one of the videos that primary spermatocytes are the first cells that undergo meiosis 1, and at this point the DNA has duplicated to have 4N the amount of DNA. So the amount of DNA is different from when we call it haploid or diploid. By diploid, we simply mean um, it's it's not from okay. What's the best way to describe haploid? It's half the normal chromosome number. Okay, it's half the normal chromosome number. A diploid, it's the full chromosome set in a somatic cell, while a haploid is half that set. But here we have 4n. What does 4n mean? 4n simply means we have duplicated the amount of DNA, because diploid simply means it's 2n. Okay, 2n is diploid, but if we duplicate this DNA, the amount changes to 4n. That's the amount of DNA. It still remains diploid, but the amount has duplicated. Okay, so they might ask you this. They usually like this MCQ. The primary spermatocyte has what amount of DNA? The amount is 4n because it is duplicated before the first meiotic division. So after meiosis 1 is complete, the secondary spermatocyte is going to have 2n the amount of DNA, although it's haploid. So it's 2n the amount of DNA because it's haploid, but it's still duplicated. The strands are still duplicated. But then those strands, as you are going into uh, the spermatids are going to have single strands each at the end of the day. That means those single strands now are just simply going to be N. That's what that means. So they usually like this question, especially the primary spermatocytes are the first, or rather, what amount of DNA does the primary spermatocyte have? It's 4N, the amount of DNA that was duplicated. Okay, so at this point, you have already said uh, you are starting with spermatogonial stem cells that give rise to spermatogonia. Then spermatogonia, we have different types, type A and type B. These ones are all undergoing mitotic division, just increasing in number, and then the actual meiosis starts all the way up to that point. So the meiosis starts primary spermatocytes, meiosis 1, secondary spermatocyte undergoes meiosis 2, and then when that completes, you end up with early spermatids. Okay, so the amount of DNA in a primary spermatocyte before it undergoes that's 4n. So at this point now we're at the early spermatids. Our meiosis is complete. The late spermatids now have to shed off the excess cytoplasm, and then they're going to undergo a few modifications which we are calling spermiogenesis. Okay, we are calling spermiogenesis. Okay, so this is just the picture, just summarizing what we're just from saying. Just another picture that will help you understand that. But what we are talking about just after that is spermiogenesis. So the process or the sequence of events that transform a late spermatid, particularly a late spermatid, into a mature sperm in the seminiferous tubules. These events are what are termed to as spermiogenesis. So from this point, from a late spermatids all the way to a mature spermatozoa, there, that's what we are calling that sequence of events is called spermiogenesis. So we have spermatogenesis. Don't get this confused. Spermatogenesis, uh, consisting of all the processes, including spermiogenesis itself. So let me just go back to this picture here. The whole process is called spermatogenesis, from spermatogonia to spermatozoa. But just here, from late spermatids all the way to a spermatozoa, that's spermiogenesis. That's the difference between the two. So in spermiogenesis, what is exactly happening? There is events that are occurring during spermatogenesis. There's the nucleus condenses. Okay, the nucleus will condense, the flagellum develops, Okay, so the nucleus, see the nucleus condensing, becoming smaller. The flagellum develops. We're having a flagella there that will help the sperm to swim. The mitochondria aggregate around the base, forming the flagellum. The mitochondria fuse to form a supermitochondrion in humans. The Golgi apparatus forms the acrosome, excuse me, and the centrioles form the annulus. Okay, and the excess cytoplasm is shed off, and it is phagocytosed by the Sertoli cells. No cell division occurs during this process, and it is the temperature-sensitive process of sperm production that requires a temperature that is slightly lower than the normal body temperature in humans, that is. Okay, so that is basically what is happening in this process of spermiogenesis, as you can see, summarized pictorially and also with these steps occurring there. So the Sertoli cells themselves that we're talking about that actually digest the excess cytoplasm, these are supporting cells whose functions include to form the blood testis barrier, which is the tightest physical barrier in the human body. Why is that so? It is the tightest human physical barrier in the human body, basically because it is responsible for protecting against autoimmune attacks. So, protecting against autoimmune attacks by the immune system of the cells. Because remember that during meiosis, you produce daughter cells that are different genetically, and the body does not recognize those cells because they have a different genetic makeup from the parent cell. Okay, so all the other cells in the body have different genetic makeup, and these are more like foreignized because crossing over has occurred and everything. So the genetic makeup is quite different there. So the immune system might recognize those as uh, foreign cells. So the reason why there is a blood testis barrier, the strongest testis physical barrier in the body, rather, it's because of protecting those uh, cells from autoimmune degradation. Okay, so now this arrangement makes sense that we were talking about. Uh, we have, the peritubular cells here, and then the basal lamina. This is inside the seminiferous tubules in the testis. So we have the basal lamina here, and then type A spermatogonia here. After the spermatogonia, you're going to have primary spermatocytes. After that, you have early spermatids, late spermatids. So all of these are just ascending up from this picture that is here. Okay, from the spermatogonia, spermatocytes, and then the spermatids, early and late. So just know them like that. You have spermatogonia type A and type B. We have primary and secondary spermatocytes, late and early spermatids. And at the end of the day, you have the mature spermatozoa. And this is represented in this picture as well. All right. So at the end of the day, Sertoli cells have functions including, like we already said, formation of the physical barrier. There's nutrition. They support and protect the developing spermatogenic cells. And then the other function is exocrine and endocrine secretions, which are androgen binding proteins (ABP), and then it also concentrates testosterone so that uh, it is aiding in the development of these cells properly. And then lastly, it's phagocytosis. Like we said in the previous slide, that the excess cytoplasm is shed off and phagocytosed by the Sertoli cells. That's one of the functions as well. So parts of a mature sperm, these are parts of the mature sperm that you're going to see. Parts of a mature sperm is going to have an acrosomal cap, that is a cap that is surrounding the acrosome. The acrosome contains the uh, digestive enzymes that help the sperm penetrate the oocyte. We have the nucleus that carries the genetic material, of course, and then the nuclear membrane containing the nucleus, or rather enclosing the nucleus. Then we have the proximal centriole, and at the end of it, or at the base, we have this uh, arrangement in the mid piece that has uh, mitochondria that help this sperm in motility. At the end of the day, there's an annulus, which we said uh, in this slide here. Where is it? Yes, the centrioles will form the annulus. That's the annulus that we are talking about. And then the annulus, from there, we have the axial filament that is in the principal piece of the tail. The tail has uh, three sections. So depending on which books you read, some of them might say we have the head, the mid piece, and the tail. Some might say we have the head and the tail, and then the tail is made up of the mid piece, the principal piece, and the terminal piece. Just know exactly what you're talking about, where it is, and what it has, and then you'll be safe. But this is the general arrangement of the parts of a mature sperm. If there is anything, we've come to the end of this particular video.